So this is the continuation or the second part of antimicrobials. So really the primary focus of that first part was to walk through both the historical contributions of different individuals with antibiotics, but then also start to look at the different mechanisms by which antimicrobials can act. So there are 6 different categories, and there are examples from each that we walk through. And now we get to some clinical considerations. And so much like we saw for disinfectants and antiseptics, there's a list of things that are probably pretty straightforward in regards to what we should consider. So readily available, inexpensive and stable, easily administered, selectively toxic. We use that term so it doesn't harm humans, but it does impact those microbes that might be making us sick. So those are all pretty straightforward. These last several we're going to talk about in more detail. So spectrum of action is described on the next slide. And then we'll talk about actually figuring out how does one come up with a dose that's appropriate for antibiotic usage or for antimicrobial usage. And so the answer to that is when we say efficacy or the effective dose, we really need to know what we're trying to achieve. Are we trying to inhibit the growth of the bacteria, or are we trying to kill the bacteria? And so there are tests that will provide the answers as to what dose will achieve those things. So that's what we're going to do. So this slide shows you what spectrum of action is, and there's 2 categories that you should be aware of. We can talk about drugs that are classified as narrow spectrum, or we can talk about drugs that are classified as broad spectrum. And so as you might guess, broad means it's going to be an antimicrobial that's going to be effective against a much larger number or greater number of different types of bacteria. And so if you look at sulfonamides, and so that's the category that we learned that targets a certain pathway of metabolism for bacteria. It's a competitive inhibitor of an enzyme that converts PABA to folic acid. And so if you look at how wide this bar is, you can see that it straddles multiple categories of bacteria from both gram-negative and gram-positive and some other bacterial types that we haven't even talked about. The same goes for these guys, these 3 bars here, streptomycin, erythromycin, tetracycline. Those are all protein synthesis inhibitors, and they are pretty wide as well. They can impact a variety of different types of bacteria, so they would be classified as broad spectrum. But if you look at this one, this is a pretty short bar from left to right, and it's only in this category of gram-negative. So if you recall, polymyxin was in the category of action where cell membranes and outer membranes are disrupted by the drug. And so gram-negative are much more affected by that. And so this would be classified as a narrow spectrum antibiotic. So there's some things that might not jump immediately to your head when we talk about broad spectrum. And so that sounds like that's going to be a really helpful antibiotic because it's useful against a wide range of antibiotics. But one of the problems with that is when you use that, not only will it negatively affect, hopefully, the microbe that's making you sick, but it can also affect those microbes that live on and in us. We call that the normal flora or the normal microbiome, normal microbiota. And so what that leads to, when you disrupt that normal flora, you can get the growth of unwanted or the growth of microbes that normally wouldn't grow as robustly, and so you can get these secondary infections. Or it can lead to what are called superinfections, or the rise of antibiotic-resistant microorganisms. And so we'll see that in the future as well. There's some slides that talk about antibiotic resistance towards the end. But spectrum of action is something that we're interested in to know how many microbes and how many different microbes a given drug is going to target. So the effectiveness or efficacy of a given drug is also something that's pretty important. And so there are 4 different tests that we're going to talk about. And so the goal for each is to understand what information is derived from a given test. So if you were to perform this test, what do you learn by doing it? And then just the basics of how it's performed. Okay. So the first test that we're going to talk about, the name of it is called a broth dilution test. So remember what broth means. It's just a liquid culture media. And so in each one of these tubes, there's broths. And what's different about them is there's an increasing amount of a certain antibiotic drug going from left to right. And so this is going to be the highest dose that you're testing. And over here, this is the lowest dose. Okay. It doesn't necessarily matter what this is, but just based on the abbreviations, this is testing tetracycline. But you could do this for any drug. If you want to do penicillin or azithromycin or Cipro or whatever antibiotic you're interested in, you could set up a series of these broths with increasing amounts of a given drug. And then what you do is, again, the term we use for this is inoculate. So that means to place a specific bacterium in here. So you can test E. coli, you can test whatever bacteria you're interested in. And once you've kind of inoculated or seeded bacteria in each of these tubes, you're going to look for growth. And so the word we used for growth in liquid cultures is turbidity or cloudiness. And so hopefully you can see in each one of these tubes, they look, I guess I should do CL for cloudy. These are cloudy. Oh, I'll write it out because I'm just realizing that clear also starts with. So these are all cloudy, but these are all clear. And so what you're looking for, this test is called an MIC test. That's an abbreviation. What that stands for, minimum inhibitory concentration. And so what this test gives you is lowest dose that inhibits growth. And so that's what MIC means. It's kind of in the name. So minimum concentration, that's lowest dose. And then inhibitory, so just realize this is not looking for death or killing of bacteria, but just inhibiting growth. So because these are clear, we can say the bacteria didn't grow. And so this tube, this would be the lowest dose that causes that inhibition of growth. So that's how a broth dilution test is performed. Another way to get the same information, so this is called an E-test. So this is the second test that we're interested in learning about. It also is an MIC test, so that's the information it provides, the minimum inhibitory concentration, but it's done a little bit differently. So this is a strip that within it is a certain antibiotic. So a strip with a certain drug or antibiotic. In this case, it looks like it's oxacillin for OX. But it's not just the drug. Instead, it's a gradient of the drug. So if you look at these numbers, big number, and they get smaller. So again, what you have is a high dose of the drug, and then you have a low dose of the drug. And then when you put this bacteria on here, and you put this strip next to it, or kind of embedded within this bacteria, what you're looking for is a zone of inhibition. So this ring, it kind of looks like an egg shape. So this clearing is where the bacteria are failing to grow, and then this creaminess is the bacteria growing. And so what you're interested in is where is the dose right here, because if you get above that dose, it doesn't grow. If you get lower than that dose, it does grow. And so this intersection that you can see on the plate is the minimum inhibitory concentration. So again, that's the lowest dose of the drug that inhibits growth. Okay, and so these are two different tests that allow you to obtain that same piece of information. So broth dilution or the E-test. The next test that we're going to talk about has two different names. So the Kirby-Bauer test, also called the diffusion susceptibility test. And what's important for this one is this is one of the tests that allows you to test multiple drugs at once. And so on the previous, I'm just going to jump back for a little bit. This was just focusing on one drug like tetracycline or oxacillin. And so obviously you could perform this with another test, another series of tubes with a different antibiotic in it. But it's this test, the Kirby-Bauer test, that allows you to, in one single test, to actually screen multiple drugs at once. So it's really helpful. It's not going to give you a dose, but instead what it's going to give to you is this bacteria, whatever bacteria you put on the plate, is it sensitive or resistant to each of these different antibiotics? And that's a really important finding to have. And the way that you do this is you put the bacteria, so that's this creamy stuff, on the plate, and then each of these little circles represents a paper disc that has been soaked in a particular antibiotic. So TE for tetracycline, AM for ampicillin, this one's gentamicin, and so on and so forth. And so these are each different antibiotics. And what you're looking for is a zone of inhibition. And as you might guess, large zone means the bacteria are sensitive to that antibiotic. So another way of saying that, when you see this big zone of inhibition, that means this antibiotic is effective at inhibiting the growth of this bacteria. So we see this nice large ring. This bacteria can't grow in the presence of this antibiotic. When you look, I'm going to choose ampicillin here. This is a smaller zone, and you can actually see these little colonies that are creeping in. So a smaller zone, what that means is the bacteria are resistant to that antibiotic. So the bacteria resist the effects of that antibiotic. The antibiotic does not work as effectively. And so in a clinical microbiology lab, if there's a patient that's sick with a potentially drug-resistant bacterium, it's really critical to find out, well, which drugs actually work to treat this pathogen. And so this test could be a really important test to determine, okay, this bacteria that's making you sick is sensitive to antibiotics A and B, but it's resistant to C and D. And so, of course, that would lead you to conclude that you would use A and B since the bacteria is sensitive to it. So this is a really important test to give you that information. But what it doesn't tell you is what dose is effective. And so that's where an MIC test, like an E-test or a broth dilution test, could be helpful. So the last test, it's related to an MIC test, but notice the abbreviation is changing. So this is a minimum bactericidal. And so this is an ending that we saw not too long ago. So side or cidal relates to killing of. And so when we talk about bactericidal, we're looking at killing the bacteria. And again, minimum concentration is another term for lowest dose. So what this test tells you is the lowest dose of a drug that kills bacteria. And the way that this is done is it's actually an extension or a continuation of that broth dilution test. So that's from the first slide, so this test. So these clear tubes are what are going to be important to input into an MBC, or minimum bactericidal concentration test. So it says clear tubes. So if you look, this says clear MIC tube. So this is, as we just mentioned, from that broth dilution test. Okay. So clear from the broth dilution test means there's no growth, but what we want to find out is, even though the bacteria aren't growing, are there any live bacterial cells present in any of those tubes? So these are the concentration of that drug. And so then what we can do is take we can take this liquid and put it onto, these are Petri plates, and you guys are starting to get more experience with those because we're working with them in our lab kits. So these are Petri plates that have drug-free media on it, so it's just nutrients. And so I'm just going to draw a couple spots in here. So there's some bacterial cells, and I'll say, okay, that one's dead, and that one's dead. But if there's still some living cells, then when that's put onto a drug-free media with nutrients, those individual cells will grow into what we call colonies. Okay, so this still has some live cells in it because it's yielding colonies, and so this dose doesn't kill all the bacteria. So same thing here, there's some bacterial cells in here, but if the dose is high enough to kill all the bacteria, the way that we detect that is when we put it onto this drug-free media with full of nutrients. No colonies means no living bacteria, and so we have killed all the bacteria. And this one has killed as well, but again, we're looking for the lowest dose. So even though this yielded no colonies, and maybe this next one did as well, this tube would be the lowest dose that yielded no colonies, or another way of saying that is the lowest dose that killed all the bacteria. So those are 4 drug efficacy tests that give you different information. So it's really important to understand what information do you need to know about this drug. Do you want to know what's the lowest dose that kills the bacterium? Well, that's the MBC test. Do you want to know the lowest dose that inhibits the growth of the bacteria? We have 2 tests to do that. Or are you trying to figure out for any one bacterium, which drugs is it sensitive to, which drugs is it resistant to? And so that's achieved through these different drug efficacy tests. So while dose is important, we also have to realize how that drug is going to enter the patient. And so these are just remind you of the different ways in which we can administer. And so obviously there are some infections that are just on the outside of the body. So it could be a topical drug. And so there are antifungals that are topical. There's also antibacterial ointments that are topical as well. And so those work just fine if it's an external infection. But if the microbe is in our body, how are we going to get that drug in there? And so there are 3 listed here to deliver through different kind of routes. Excuse me. So the first is oral, which obviously you just swallow the antibiotic pills. And that is helpful because you don't have to have any specialized delivery. You just have to be able to swallow pills, or even for younger kids, it can be a liquid suspension of the drug that tastes like bubble gum or whatever. So that's good. The problems are when you take something orally, it takes some time for it to actually reach the bloodstream. And so this picture over here is looking at the relative concentration of these different delivery methods over time. And so time 0, which would be here, is when you actually administer the drug. So in this case, when you swallow the pills, it's going to take some time before we get that increase. And it's not a real fast increase. It's slow over time because it has to go to the stomach and then to the intestine, and then you absorb it. You also have to rely on the patients taking the drugs in the way that you're supposed to. So are they taking it twice a day? Are they taking it with food so it helps with absorption, etc.? So patients don't always follow the prescribing instructions, which can be problematic. Intramuscular is not a real common way for antibiotics. It's usually oral or IV, but this is another route. So anyways, it's via an injection, and it just goes into the muscle. And so you can get a pretty rapid increase in concentration, but it's going to diminish over time. Whereas an IV, if you deliver it directly into the veins, then what that does is it gives you a very rapid infusion of the drug, and you can maintain that very specific dose by continually dripping it in through that IV. So that can be really helpful if it's a potent infection that you need to try to get under control really rapidly. You don't want to wait several hours before the effectiveness kicks in. In terms of adverse effects, there's different ways that we can look at this. We can talk about toxic effects of the drug itself. And so, we try to achieve this selective toxicity in many of these drugs, but there are certain tissues or cells that might be more affected than others. And so there can be toxic side effects of these different antibiotics. So that's something that we're concerned about for sure. There's something that we know it happens with different drugs and where people can develop allergic reactions to drugs. So although the drugs themselves may not have toxic effects, your immune system is responding to it, and then that response leads to changes in your physiology that can range in severity to even life-threatening if someone has an anaphylactic shock reaction to something, which can lead to constriction of the airways and the inability to breathe, etc. So those can range in severity, but they're a definite concern. And something we already touched upon, if you're using a broad spectrum antibiotics in particular, and especially if you're using them over a long duration, you're really going to kind of wreak havoc on those normal microbiota that live in the intestine, and that harbors trillions of different bacteria that are present. And so if that gets disrupted, that could lead to potential consequences of secondary infections growing, where you can get this yeast candida that grows, or C. diff, which is another organism that can be found in the intestine. And it might be able to grow better because it's not susceptible to some of those antibiotics, and so you can get these secondary infections that occur. And we see this in hospitalized patient, as you might guess. It's if someone's really sick and requires an IV antibiotic, they're likely going to be hospitalized. And so we'll talk about what are called nosocomial infections, or healthcare acquired infections, in another lecture later on. But it's definitely a concern that we have when we deliver these drugs. So resistance is something that I've touched upon. And what we mean by resistance is that the bacteria are able to grow in the presence of that particular antibiotic. And we can be a little more specific. It may be that the drug still works, but it's reached something that's called clinical resistance, where the lowest dose that you need of that drug, so that minimum inhibitory concentration, MIC, of a particular drug is too high to be used to be safe. And so that what we mean here is, so in the patient, we would have to use such a high dose that it's no longer safe anymore. So that would be then reaching this clinical resistance. So there's kind of two lists that we want you to know in regards to resistance. So this first list is, how can bacteria change or evolve over time so they actually, at a genetic level, so in their DNA, how are they going to get this new trait? So how do they acquire this resistance gene? And so there are four explanations. You can argue it's two, but so the first is mutations occur, and I'm going to show you a video of this in just a second. These are all something that we're going to talk about in another lecture. They're classified as one group. This is called horizontal gene transfer. Horizontal gene transfer. And so there are three of these horizontal gene transfer mechanisms that we know to exist within bacteria, and they have their individual name. So transformation, transduction, and conjugation. So you don't have to know the specifics of these right now, because we're going to cover those in the genetics lecture, which I think is the next one, so lecture 9, I believe. So you're going to get more information, but right now, what we want you to know is those three horizontal gene transfer mechanisms, transformation, transduction, conjugation, along with mutations, is what explains how bacteria can evolve over time and gain new genetic material, including resistance genes, so antibiotic resistance genes. The next list, and the reason I'm teasing these apart is these are frequently confused, and so I'm just going to repeat what this list is talking about. So if the question is asking at the DNA level, or at the genetics level, or at the gene level, how does a bacteria acquire resistance, the resistance gene, these are the four answers. But another way of looking at it is, okay, so if a bacteria does get an antibiotic resistance gene, how is it actually allowing the microorganism, the bacteria, to survive in the presence of an antibiotic drug? And so this is looking at what is that gene actually doing at a cellular level to impart that resistance. So there's a couple, there's 4 ways listed here. So the first is the gene leads to the production of, say, an enzyme that actually inactivates or destroys the antimicrobial agent. Okay, so that is right here. The numbers don't match up, I apologize, but so inactivating enzymes. So that resistance gene is allowing the bacteria to make something like an enzyme that deactivates or inactivates or destroys the actual drug. So even though the drug is administered, the bacteria break it down. The second is altering the permeability. So remember, bacteria have this outer layer on it that's composed of, well, it depends on the bacteria, but some have an outer membrane and peptidoglycan and a cell membrane. Others have just peptidoglycan and a cell membrane. But it is a barrier, and so if bacteria can block the entry of that drug, that can be really effective. So if that antibiotic can't enter the cell, it can't act. Or if it does enter, but the bacteria actively pump it out. So these, it can be argued it's 1 or 2 mechanisms. This picture shows it as 2 separate things, but inability to enter or active export. But they both fit under the larger category of not allowing the drug to enter the bacterial cell. Another mechanism here says altered target site. So that's what's being shown here. So what we're talking about is that if the drug targets a specific thing in the bacterial cell, if that target is changed so that it still functions in the bacterial cell, but now the drug can't act upon it, that can be another way of providing resistance. So here's the antibiotic, and here's the altered target molecule as it shows there. And then the last one is just the replacement. And so I'll just give you an example. So this is one of those metabolic pathways that we were talking about. So bacteria take PABA and they make folic acid. And that's important in making nucleotides and then making DNA. And so this is what the drugs do. They block the enzymes that are responsible for converting PABA to folic acid. If bacteria found a new way to acquire folic acid, so say they started to import it from their environment or whatever it is, so this would be a replacement of the sensitive pathway. So if they could find a new way to get folic acid, then they could pick up and start making those nucleic acids again. So that's what we mean by a replacement of that sensitive pathway. So any one of those mechanisms could explain how a bacterium could survive in the presence of a particular antibiotic. So the development of resistance is something that we're really concerned about. And so this diagram is one that helps explain how resistance can occur over time. And so this is a Petri plate with a population of cells with varying sensitivity to a drug. So sensitive cells versus drug-resistant cells. And one thing about resistance is the more we use an antibiotic, the faster that we're going to see resistance evolve. And so not surprisingly, in hospitals and nursing homes where people are going to be on antibiotics more regularly, we actually see the evolution of drug-resistant bacteria. And one big problem is the development of multiple drug-resistant strains. So in one bacteria, they're resistant to multiple types of antibiotics at the same time. So in the news, these are called superbugs. That's not the best term, but if you see that term, it's just a multi-drug resistant bacteria. So when there is exposure to the drug, obviously the sensitive cells are going to not grow or die, but those that are resistant can still grow in the presence. And so they'll start to increase in abundance in that population and kind of take over. And so over time, as that antibiotic is used, we see kind of a shift in the population to drug resistant. So this is a pretty amazing video of an experiment that was done at Harvard. So I'm just going to show it to you. I think it's 2 minutes long. It's embedded in this. So I may voice over it, but...
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
So that video kind of helps illustrate this fact that as a drug is used and as the concentration increases, it only takes one mutation. In that video, it actually showed multiple mutations occurring over time, these kind of step-by-step changes that allow a bacteria that wasn't resistant at all, but now it can survive in something that's 1,000 times more concentrated than the original antibiotic. And so that's the problem that we encounter. The more that we use an antibiotic, the more that the bacteria is exposed to it, it provides the selection needed for that mutation to take hold and grow and spread within a population. So it's a growing concern across the planet. And so one thing that we want to think about is how can we avoid or delay the resistance to antibiotics. And so obviously making sure that you're using the right dose and the right duration so that patients are actually take their entire course of antibiotics. So that's frequently part of the instructions. And so it's important to teach patients of the importance of this so they're not contributing to the development of antibiotic resistance. But then also to limit the use of antimicrobial agents to only those necessary cases. So we tend to be a culture where we want to have some prescription, and so people actually ask for antibiotics when they're not warranted, and it's important for healthcare providers to inform them and provide them the information necessary so they understand the reason why that's not a good choice. And it also is just important to know that most antibiotics are not actually used for medicine, but rather they're used in agriculture, and again, not for veterinary purposes, not because livestock are sick and we're treating them. This is just in the feed to allow the livestock to grow. What's been demonstrated is that in chickens, they just grow faster, so they get bigger more quickly when there are antibiotics in the feed. And so that's a business decision, but it's not a great public health decision because we know that there are antibiotic-resistant strains of bacteria that are developing because of that practice. Using more narrow-spectrum antibiotics or using antibiotics in combination is helpful, and then also just developing new drugs in these categories of antibiotics that we have. So these are all working, hopefully working together to help us avoid, or if we can't avoid, at least delay antibiotic resistance.